Hello and welcome to all my dear students. So today we are going to start with the lecture number 3 of the chapter Cell, the Unit of Life. So far beta, we have discussed the detailed structure of prokaryotic cell, and I think so today we have to start with the detailed structure of eukaryotic cell, in which we are going to discuss first the layers of eukaryotic cell. So there are two possible layers, one is cell wall and the other is cell membrane. Then followed by the cell organelles we have to discuss. So without wasting time, let's directly start with the structural organization of a eukaryotic cell, starting with the outermost layer, that is cell wall. However, you all know beta that presence of cell wall is not a feature of all organisms. Some organisms like plants, some protisteans like diatoms, dinoflagellates, fungi, algae, they have cell wall, but organisms like animals do not have cell wall. So in them, the outermost membrane is plasma membrane. However, in these organisms like plants and fungi, the outermost layer in their cell is cell wall. So presence of cell wall is a feature of, the first point what are you going to write is? Yes, very good. That is presence of cell wall is a feature of plant, fungi and some protisteans, right? So this is the first point that you have to remember. Second beta, it is a dead layer. Dead means it will not act as a barrier for the movement of material, and yes, it is a fully permeable layer through which any material can easily move in and out. Right? Now when we talk about it, what this layer is made up of, so like you have seen in case of bacteria beta, this layer is uniquely made up of peptidoglycan murin. However, in eukaryotic cell wall, there are different composition depending upon the type of the cell. Like fungal cell wall is made up of chitin, which is a polymer of N-acetyl glucosamine. Then algal cell wall along with cellulose and insoluble polysaccharide. It also has some other crystals of galactans and mannans, whereas typical plant cell wall has cellulose, hemicellulose and pectin. So depending upon cell to cell, the composition varies. So now next you're going to write is depending upon the composition, right? So depending upon the composition, there are three types. First of all, fungal cell wall. Now beta, what this fungal cell wall is made up of? Yes, very good. So this fungal cell wall is made up of chitin, right? Which is actually a polymer of NAG. Second is algal cell wall. Now, algal cell wall is made up of cellulose. Along with cellulose, it also possesses sugar components like galactans, mannans and crystals of calcium carbonate, right? Crystals of calcium carbonate. Now, moving with the third, that is the most important, plant cell wall. So, what plant cell wall is made up of? Yes, very good. Cellulose, hemicellulose and pectin. Correct? Yes or no? So these are the different types of cell wall depending upon the composition. However, if you want to see the types of cell wall on the basis of origin, that how they have developed and what is the time of the development of cell wall, then again we have two types, primary cell wall and secondary cell wall. So first listen to me carefully and then try to understand it. Okay? So basically, beta, if a plant cell is young, immature, then around that immature young plant cell, the wall which is present is known as cell wall, primary cell wall. This is thin, elastic, living layer capable of expanding because it has proteins like expansins and extensin which helps this primary cell wall to grow. Now, with time, as this immature plant cell matures, this extra primary wall expands and a gap is created between primary cell wall and plasma membrane, which is then occupied by deposition of new rigid thick secondary cell wall, dead cell wall, which is called as secondary cell wall, right? So depending upon the origin, we have two types of cell wall. Yes, depending upon the origin of cell wall, beta, we have two types. Number one is primary cell wall and second is secondary cell wall, right? So primary cell wall, beta, it is found in immature cell. It is found around in a young and immature cell, whereas secondary cell wall is found around mature cell, found in a mature plant cell. I'll draw it with the help of a diagram also. I'll try to explain with the help of diagram as well. But first write the points. Now, primary cell wall, as I told you, is thin, living, very important, capable of growing, that means it is flexible, can expand. However, here, this is a thick layer. Thick means it is several layered. It's not just one layered. Rather, it is several layered, followed by being dead in nature. Now, since it is dead, so it cannot expand. So, no expansion, you can say simply, it is non-flexible. Right? Clear to everyone? Now, let's try to understand this with the help of a simple diagram. Okay? Chalo. So beta, first over here, I am going to draw an immature plant cell. So first we are going to draw over here an immature plant cell which is just having one layered cell wall, that is primary cell wall. Now as I told you that this primary cell wall is thin, flexible and capable of further growth. So this is my primary cell wall. Right? This inner one is the cell membrane. It is having cytoplasm, vacuole, etc. Now, this is beta immature cell, right? Young, immature means young plant cell. Now, with time, this cell is going to mature, yes or no? This cell is going to grow in size and will become fully mature so that it can further perform functions and can further divide. So what happens beta? On maturation, basically this primary cell wall expands Now, because of the expansion of primary cell wall, there is a gap created between it and the plasma membrane. And now in that gap, there is the deposition of new layers. And these new layers are known as secondary cell wall. What are these known as? These are known as secondary cell wall. Correct? Chalo. So now my primary cell wall is expanding, and due to the expansion of primary cell wall, a gap is created. Now this is your primary cell wall which has got expanded. This is your cell membrane or plasma membrane, and now you can see a huge gap in between them, and in this gap there will be the deposition of new beta secondary cell wall. Right? So here you can see the deposition of new secondary cell wall, and not one just layer. The secondary cell wall is several layered thick. Every layer is placed one inside the other. So all these are secondary cell wall, which all these are laid one inside the other. Correct? Now with time, imagine I give you a rubber. If I give you an elastic and I ask you to expand it, a time will come that the elasticity property of that rubber will exceed the limit, and now your elastic will burst, or you can say it will break. Right? Same thing happens over here. A time comes when primary cell wall further does not expand, and due to continuous tension, that primary cell wall breaks and it vanishes, and now the only cell wall left is thick, dead, tough layer called as secondary cell wall. Right? That is why we say that cell wall is dead layer, tough layer and thick layer. Why? Because we are talking in terms of a mature cell. And of course, a mature cell is having secondary cell wall, which is dead, which is not elastic, which is not capable of further growth. Rather, it is thick, and yes, it is resistant and provide strength and rigidity. Right? Clear to everyone? Okay, very good. Plus one more thing, beta. This secondary cell wall is laid inner to primary cell wall, not away or not exterior to primary cell wall. So this is just an example of showing intrinsic growth. You know that all living organisms without any exception shows intrinsic growth. Intrinsic is basically internal growth, where the whatever new growth will take place inside the body, inside the cell, not outside it. So here already the cell is this, already primary cell wall here. Now whatever new deposition has to take place, that should take place inside the cell, not outside the cell. So whatever secondary cell wall deposits beta, everything is done inside the primary cell wall, not outer to it. So one more point over here, you can write deposition of secondary cell wall is an example of intrinsic growth. Right? It is an example of intrinsic growth. I hope now this is clear. Now with time, primary cell wall due to continuous expansion will break and it will vanish. Now beta, it's simple, like imagine if I ask you to build a wall, right? And I give you blocks, that means I give you bricks. And I ask you, okay, you have 100 bricks, build a wall. So what will you do? You will just keep all the bricks one above the other and say that, okay, ma'am, the wall is ready. But will that wall be strong enough to bear any kind of force and pressure? No. Why? Because you have just kept the bricks one above the other. You have not glued them. You have not cemented them. So still you might have placed blocks one above the other, but it's weak. If I'll just push it once, all the blocks will fall down. However, if you apply some cement in between them, if you glue them, then that wall will become rigid, full of stamina, full of strength. Right? Similarly over here also, beta, you know, all multicellular organisms, we are made up of cells, right? Correct? But are all the cells kept like that? Are all the cells simply associated with each other without any connection, without any cementing substance? No. There has to be a cementing substance between every cell so that a body can be a firm structure. So yes, in case of plants, beta, every two cell is glued with the help of a cementing substance called middle lamella. Very good, everyone. So next you are going to write about is middle lamella. Now what is middle lamella? It is a cementing substance that keeps the two cells together. That keeps the two cells together. Right? So basically it's a cementing substance that keeps the two cells together, and now it is made up of calcium and magnesium pectates. It's made up of calcium and magnesium. Now, see, suppose this is my one plant cell, and then over here I placed another plant cell. Then between two cells there will be middle lamella. Okay, it's just like that there was a plot. I had a plot and I wanted to build 4 rooms. So what I did, I basically built 4 walls separating my whole plot into 4 rooms. Dekho, I wanted to say this. Suppose this is the room. So this is the plot. This is a piece of land that I had, and now I wanted 4 rooms on this plot. So what I will do, I will build 4 walls. Yes, this is 1 room, 2 room, 3 room, 4 room. Is it okay? Sahi hai itna? Will I be able to live in this home? Why won't I be able to live in this home? Because I have not connected the rooms. I have simply built the walls without adding any window or doors. And you know, without windows and doors, how will I move from one room to another? If I enter in one room, I will remain there forever, because everywhere there is a wall. And then how will I move out, or how will I connect to my other rooms? It's completely useless. Have you seen any home without doors and without windows? No. Have you ever seen any home where there is only walls all the 4 sides without any door, without any window? Not possible. Because without doors and window, how will you communicate to the outer world? How will you get attached to the other rooms of the home? So these are very, very necessary for keeping an interaction. Similarly, beta, in plants also, if there are 2 cells and if in between them I completely fill up the cement, then how are they going to talk to each other? How are they going to exchange their cytoplasmic material? For that, for the exchange of cytoplasmic material, we need to establish some living connections. Yes, very, very good. Such living connections are known as plasmodesmata. Very good. So, the next is plasmodesmata. Now, what is plasmodesmata, beta? These are the living connections between the two cells for the exchange of, I'll show it with the help of the diagram also, between the two cells that allows the exchange of material. Yes, correct everyone, that allows the exchange of material between them. Like see over here, let's draw, let's try to understand with the help of an example. Now this is my one plant cell, right? Now the plasma membranes are always connected, because to establish plasmodesmata we need to have connected plasma membranes. Now these are the plasma membranes connected, this is the cell wall, right? And now between the two cell there is a cementing substance called middle lamella. So this is plasma membrane, cell wall, middle lamella, and now here lies your cytoplasmic connection, living connection called, yes, very good, plasmodesmata. Right? Now, this plasmodesmata helps to allow the exchange of material adjacent to cells, and this plasmodesmata beta, plasmo word has come from plasma membrane, because to make this connection we need the linings, we need the living linings of plasma membrane, plus desmota word has come up from desmotubule. And these desmotubules are made by endoplasmic reticulum. These two together makes the plasmodesmata, which is a living connection for the exchange of material adjacent to cells. Correct? Got my point? Now, that's it for cell wall. Proceeding with the next, that is cell membrane. Now, inner to cell wall is cell membrane, which is found in all cells, be it plant, animal, fungal, any cell. All the cells have at least this membrane called cell membrane. Unlike that of cell wall, it is a living membrane and selectively permeable, right? That means it will allow only selected molecules to move in and out of the cell. One thing. Second thing beta, structurally the cell membrane is similar to prokaryotic cell membrane. Now listen to me carefully. There are two types of cell, prokaryotic and eukaryotic. These two cells are highly different from each other, except the structure of plasma membrane. This is the only thing which is common in between two. In both prokaryote and eukaryote, membrane is made up of lipid bilayer along with proteins embedded. Right? This model was very well explained by Singer and Nicholson in the year 1972 while working on human RBC under a very magnified electron microscope. Yes, got my point? Chalo, let's write. So, living membrane, first point. Second, it is a selectively permeable layer. Third, structurally similar to prokaryote. Structurally similar to beta prokaryotic cell. Yes, the model explaining the structure was proposed by Singer and Nicholson in the year 1972, and the name of the model was beta fluid mosaic model. Right? So what is the name of the model? It is actually fluid mosaic model. Understood? Chalo, very good. So model was proposed by Singer and Nicholson in 1972, and yes, the name is this while working on human RBC. While working on human RBC under well designed electron microscope, where they found that human RBC consists of 40% lipid and 52% protein. Correct? Yes or no? Now, how this lipid and proteins are present? So that we are going to study with the help of a diagram. I'll draw a diagram, I'll explain it, I'll write the points so that as and when you get the time, you also complete your notes. See students, completing notes is very, very important. Only listening to the lecture but not writing it, not learning from it is not going to help you. Correct? So if you really want to improve yourself along with the listening to the lecture, writing is also very, very important, right? So yes, you make sure that you make your notes as and when you get the time. Chalo. So now we are actually better going to start with this 40% lipid arrangement and 52% protein. Let's see that how this arrangement is. Okay. Suppose this is my human RBC. What is the shape of a human RBC? Biconcave, disc-like, spherical, like this. Now, since you know human RBC is an animal cell, it will have only one layer that is called as animal membrane or cell membrane around the cell, whose structure we have to understand. Now, beta, whatever material or whatever region is outside this cell membrane is known as extracellular region, and whatever is present inside the cell, this is known as intracellular region. Now students, both extracellular and intracellular, they are aqueous in nature. That means they have water. It's not vacuum. It's rather filled with fluid. Fluid means they both are aqueous environment. Aqueous means watery, fluid. Right? I hope this much is clear to everyone. Now, when these two people, Singer and Nicholson, they tried to understand the structure of cell membrane, and so when they used human RBC for that and kept it under their highly magnified designed electron microscope, they saw that this membrane appears to be like two layers, one above, one below, and both the layers are made up of phospholipid. So it is like this how the membrane appears to be, right? So yes, this is how membrane beta appears to be. Correct? This is the upper lipid layer, phospholipid layer. So membrane is not single layered. Rather, it is present in the form of two lipid layers. And this is the lower phospholipid layer. Correct? So, according to fluid mosaic model, plasma membrane is represented by two layers of phospholipid, one above the other. Now, what this I drawn? Lipid को ऐसे कैसे बनाया है? So let's try to understand one molecule of each lipid layer. That means this. Take out this outside. Now here you see I have drawn a circle and two legs. So beta, this circle is actually called as head and these two legs are called as tail, right? So each lipid molecule in the bilayer of plasma membrane is represented by a head and a tail. This head is made up of hydrophilic polar saturated hydrocarbon. Hydrophilic hydrocarbon, whereas tail is made up of hydrophobic, means non-polar saturated hydrocarbon. Now, hydrophobic, hydrophilic, do you have any idea what these two terms actually stands, beta? Hydro means water, philic means loving, phobic means going away from, darna, scared of. So this head is basically beta hydrophilic in nature. That means it loves water. Hydrophobic tail means tail is scared of water. Now, you must have seen that how have I arranged the plasma membrane. I have arranged the two layers of the membrane in such a manner that head is facing the aqueous environment from both the side and tail is getting protected from the aqueous environment by the head. So yes, you have to arrange the lipid bilayer like this only because head is hydrophilic. It can stay in contact with the aqueous environment, but tail being hydrophobic in nature, it cannot stay in contact with aqueous environment of both extracellular and intracellular region. That is why tail has to be projected inside and head has to be projected outside. Correct? Got my point, everyone? So what is the next point that you are going to write over here? The next point you are going to write over here is head of the lipid molecule is projecting outside, protecting the hydrophobic tail. Protecting the hydrophobic tail from the aqueous environment. Right? Clear? Understood? Okay. Very good, everyone. So this is how the lipid is arranged. Now, beta, on this phospholipid bilayer of plasma membrane, proteins are also found attached to it. Now, proteins can be either external or they can be internal. So, depending upon the location, proteins which are present on the membrane, they can be of two types. So, next point that you're going to write is depending upon the location of protein on the membrane, beta, they can be of two types, either intrinsic, also called as internal protein, or extrinsic, also called as peripheral protein. Right? Now, what is internal protein, beta? The protein which is deep embedded inside the membrane such that it is not easy to remove. Whereas extrinsic proteins are those which are just present on the membrane, both either on the upper lipid layer or lower lipid layer, in such a manner that they're very easy to remove. You can simply remove them either with the help of a blade or a knife, right? So what is intrinsic protein, beta? Proteins that are embedded. Proteins that are difficult to remove as they are embedded in the plasma membrane. Right? Clear to everyone? Whereas external or peripheral proteins are those which are superficial, means simply present on the lipid layer, not embedded, hence they are easy to extract either with the help of simple blade or knife. Correct? Chalo. So this I have told you the two types of protein theoretically. Let's try to understand with the help of the diagram also. Further, beta, intrinsic protein can be of two types. Either partially embedded or totally embedded. Now what is this? Everything I'll explain you in the diagram. So let's go back to the diagram portion. Now, suppose this is my lipid bilayer. I'm drawing in the form of lines. I'm not drawing the proper lipid molecules, but you already know this much. So I think I further need not to, draw the diagram properly. So this is my lipid molecule, lipid layer. Now on this, my proteins will be kept. Now see, this is the upper lipid layer. This is the lower lipid layer. Lower lipid layer encloses the intracellular environment, right? Whereas upper lipid layer encloses or faces extracellular environment where both are aqueous in nature. Now, some proteins are present simply on the membrane like this. Not inside the membrane, rather simply on the membrane. So, all these such proteins are called as extrinsic protein. Correct? Whereas, some proteins are embedded inside. Now, depending upon how much they are inside the membrane, either they can be partially inside the membrane like this, or they can be totally inside the membrane like this. So this is total intrinsic protein, right? Whereas this is partially embedded intrinsic protein, right? I hope with the help of the location you can very clearly make out. Now further, beta, this intrinsic protein, this totally intrinsic protein can be of two types, as carrier, totally intrinsic protein, and tunnel, totally intrinsic protein. Now, what is this carrier? What is this tunnel? Let's try to understand. So, beta, I hope this diagram is clear to everyone. Chalo. Now, our focus is going to understand the types of totally embedded intrinsic protein. Types of totally embedded intrinsic protein. Correct? For this, again, we need the lipid bilayer. Now, there are two types, as I told you. This is one type of intrinsic protein, and this is the second type of intrinsic protein. A, B. Now, one beta A is known as carrier protein. This is actually solid from inside and it is specific in nature. Now, what is solid? What is being specific in nature? Can you just little bit elaborate for us? Yes, of course. See, can you see this protein, beta? This is a protein which will help in the movement of material, but it is solid. That means directly substances cannot pass through this protein. ऐसे कोई भी material pass नहीं करेगा because it is solid from inside. Then how the materials are going to pass through it? So beta, basically these proteins have some specific binding sites called as receptors, right? They have some binding sites called receptors which are specific to specific molecule. For example, this receptor has the capacity to bind with maybe sodium ion. So, if suppose this sodium wants to come inside the cell, so directly but it cannot pass the membrane, you know, because membrane is hydrophobic in nature and this is a charged particle and charged particle cannot cross the hydrophobic membrane. Rather, it needs the help of some proteins. So, if it takes the help of A, that is carrier protein, so it will bind to its specific receptor. Once sodium has bounded its specific receptor, then now this carrier protein beta staying in its location, अपनी जगह पर रहकर, it will turn 180 degree. And now, if it is going to turn 180 degree, then sodium from outside will come towards inside. ठीक? अब ये सोडियम इससे detach हो जाएगा. Now this sodium will detach, and once this detachment occurs, now this protein will go back to its original location. Right? फिर ये वापस जाके अपनी ओरिजिनल लोकेशन में जाएगा and like this the transportation of material is done with the help of solid specific totally embedded protein called carrier protein, right? So what this carrier protein is doing, it is simply having specific receptors to which molecule binds, and then it rotates 180 degree to deliver the molecule, and then it rotates linearly 180 degree to deliver the molecule on the other side. Clear? Yes or no? Have you all understood till here? Now, on the other hand are the tunnel proteins. Beta, these tunnel proteins are rather hollow. They're simply like gates. They're simply like open gates through which pores. Tunnel proteins are basically, beta, they are also called as pores. इनको हम क्या कहते हैं? These are also known as pores. Why? Because beta, they are hollow from inside. And since they are hollow, so through them they don't want to, there's no need for them to move or to change their location. They can simply move, they don't want to move anywhere. They're simply hollow from inside, and through them material can directly pass. अंदर से बाहर, बाहर से अंदर is very simple. Here it is not that simple. Why? Because it is solid from inside. It's not like in any normal opening. So here the protein has to rotate 180 degree. Here protein need not to rotate at it as it's completely open, and through them any material can easily pass. Like for example, aquaporins. Aquaporins means the porin, the tunnel protein which allow the movement of hydrophilic substance like water itself. Right? Got my point, everyone? So I hope you all have understood the functioning and the structure of plasma membrane. Now beta comes to the next portion, that is the function of plasma membrane. If I talk about the function of plasma membrane, students, then first and very important function is bulk transportation of material. Bulk transportation of substances. Now, this transportation can be done in two ways, either by spending energy or without spending energy. That means active and passive respectively. Now beta, when the movement of substances is done along the concentration gradient, so for that movement no energy will be required. However, if the movement is done against the concentration gradient, then of course energy expenditure will be required. So passive means no involvement of energy. Why? Because movement is along the gradient. Right? Whereas for active, what will you write? Energy is required as movement is done against the concentration gradient. Now, beta, apart from transportation, the next important function that plasma membrane performs is helping in cell division, cell growth, secretion, etc. So it also helps in cell growth, cell division, and secretion, etc. So all these are also the important functions performed by plasma membrane. I hope you all have understood the two layers of eukaryotic cell, cell wall and cell membrane. Now, when you come inside these envelope, then you find a fluid, that is cytoplasm. You know, cytoplasm is the main arena for all the cellular activities. Plus, unlike that in prokaryote, the cytoplasm was continuous, not separated into further, membranes and all. As a result, in prokaryote, no membrane-bound organelles were reported. But here, in case of eukaryotic cell, compartmentalization is seen in their cytoplasm. That means membrane-bound organelles are found. So one by one we are going to discuss all the membrane-bound cell organelles, starting with endomembrane system. This endomembrane system, beta, comprises of set of cell organelles whose function are intercoordinated, interdependent on each other. And it comprises of ER, Golgi body, lysosome and vacuole. Right? So now we are going to start with the concept of endomembrane system. The system basically, beta, comprises of set of organelles whose functions are coordinated. Right? So it comprises of set of organelles whose functions are interdependent, or you can say coordinated. Right? Now, the four cell organelles which are the part of endomembrane system are ER, followed by Golgi bodies, followed by lysosomes, and last is vacuole. Right? So these are the four cell organelles. All of them, beta, they are bounded by single membrane. How many membranes are present around them? Yes, they are just bounded by single membrane. Now, first you have to understand, see, one by one we will deal in with all the four cell organelles, but first you need to understand that why and how are their functions coordinated. So here I am just going to give you a simple explanation, and remaining explanation we will do as and when we will proceed with the topics. Correct? So beta, see, ER is actually responsible for producing the two most important macromolecule, that is lipid and protein. Correct? अब होता क्या है, ER is producing lipid and protein, but this lipid and protein is not ready to be utilized. It's simply like there's a farmer who is growing crop on the field. So farmer is like ER, who is responsible for producing the crop, that is protein and lipid. But can you use that crop directly to your home? Or first those crops need certain modifications, need certain processing and packaging before you use them at home? Yes, they need certain modifications. So same thing has to happen over here also, beta. ER may synthesize protein and lipid, but this protein and lipid is right now not capable of getting utilized anywhere, either inside the cell or outside the cell. Actually, it needs modification, it needs changes, and for those changes, now these proteins and lipids will be sent to second cell organelle, Golgi. What Golgi does? Beta, it basically modifies the protein and lipid generated by ER. ठीक? So it's like the factory where you sent your crop for getting modified. Now all those modification is done. एक बार जैसे modification हो जाएगी, now these proteins and lipids are ready to be utilized, and from there comes the next organelle, which is lysosome. Right? Now this lysosome arises from the packaging of Golgi body, The Golgi body directly give rise to lysosome. So lysosome, it arises from the packaging of Golgi body. Right? Yes or no? And now, what is the role of lysosomes? Lysosomes are responsible for the digestion, right? These lysosomes are basically responsible for the digestion of material. So at last, these lysosomes, after digestion, whatever undigested portion is left gets temporarily stored in the vacuole. So like this, all four are functionally coordinated. एक बनाएगा, दूसरा तैयार करेगा, तैयार होने के बाद नया cell organelle बनेगा, and now whatever this new cell organelle is going to function and so-called waste is generated, that waste gets temporarily stored in the vacuole. So this is what endomembrane system is all about. So now this is, I have just given you a gist of endomembrane system. However, we have to study every cell organelle in detail, but that we can do tomorrow also. So that's it for today. Today you have discussed the structure of cell wall, cell membrane, and you have understood the concept of endomembrane system also. But the details of endomembrane system dealing in with all these cell organelles we will do in our next class. I hope you all have understood. If yes, then this is the question. Which year was fluid mosaic model proposed? So answer is A. Yes. Chalo. So with this, we come to the end of today's lecture. I hope you all liked it, enjoyed it, and keep in touch with all the lectures which are given to you on daily basis. Please do not ignore them. And if once you have started watching, just stay connected with the series. Every day new lectures will be posted. So if you really want to make the rhythm and wants to study with us, stay connected, stay tuned. Keep working hard. That should be the only motive of life. So thank you for watching me. Bye bye everyone. Good.